In this lesson, we'll cover the concept of pharmacodynamics. So while pharmacokinetics describes the actions of body on the drug, pharmacodynamics describes the opposite. In other words, in the study of pharmacodynamics, we look at what a drug does to a body. So when a drug enters your body, it then starts interacting with cell receptors, which in turn leads to a formation of signal. And this signal, through series of different reactions, ultimately results in some biological effect. So for example, the signal can tell DNA to stop replicating. Cells in our body have lots and lots of different receptors, which when stimulated can produce very unique responses. The receptors that have the most therapeutic relevance can be divided into four types. Number 1, ligand-gated ion channels. Number 2, G-protein coupled receptors. Number 3, enzyme-linked receptors. And number 4, intracellular receptors. So now let's talk about them in more details. Let's start with ligand-gated ion channel, which is probably the easiest one to describe out of the four. And just a quick reminder, ligand generally refers to some molecule or an ion. So this channel has the ligand binding site, and when the ligand binds to it, the channel opens very briefly, which allows ions such as sodium, potassium, chloride, calcium, etc. to pass through the membrane. And that's all it is to it. Next, we have G-protein coupled receptor, also known as 7-transmembrane receptor, and this is because it passes through the cell membrane 7 times. So these receptors are composed of 3 subunits, alpha, beta, and gamma, all together known as G-protein. In its inactive form, the alpha subunit has GDP attached to it. However, when ligand binds to the receptor, the affinity for GTP increases, so then GTP replaces GDP. This in turn causes the alpha subunit to dissociate from beta-gamma complex, and then both of these complexes go to interact with other enzymes or proteins, which they can alter and regulate, ultimately leading to some kind of a response. Now there are 3 kinds of G proteins that are important to remember. These are GS, GI, and GQ. First, GS is a stimulative G protein that activates an enzyme called adenyl cyclase, which produces cyclic AMP from ATP. Cyclic AMP is a very important second messenger. The second kind, the GI, is an inhibitory G protein which inhibits adenyl cyclase, thus lowers levels of cAMP in the cell. The last one is GQ, which activates class of enzymes called phospholipases C. We'll refer to them as PLC. Now PLC produces 2 second messengers. First, diacylglycerol, which we'll refer to as DAG, and the second one, inositol triphosphate, which we'll refer to as IP3. Now DAG, just like cAMP, leads to different responses through activation of protein kinases. However, IP3 produces various responses by mediating intracellular release of calcium. It's also important to remember that G-protein coupled receptors, as well as most enzyme-linked receptors, have ability to amplify signals that they receive. So for example, just one stimulated G-protein receptor can activate many adenyl cyclases, which will result in more cyclic AMP molecules produced and ultimately amplified response. Now let's move on to enzyme-linked receptors. These receptors, just like G-protein receptors, have extracellular binding site where ligand, typically hormone or growth factor, can attach and thus stimulate enzymatic activity inside the cell. Most enzyme-linked receptors are of tyrosine kinase type, which simply means that they display kinase activity and that there is a amino acid tyrosine involved in that. So the way it works is that when ligand binds to 2 of these receptors, it causes conformational change that results in aggregation of both receptors. Once the dimer is formed, the tyrosine regions get activated and cause ATP to become ADP, which results in autophosphorylation of the receptors. Now, once each tyrosine picks up phosphate group, different inactive intracellular proteins come up and attach themselves to phosphorylated tyrosine. This in turn causes conformational change in the attached protein, ultimately leading to a cascade of activations that produces cellular response. Next, and the last type of receptor that I want to talk about is intracellular receptor. Unlike the other three, this receptor is located entirely inside the cell rather than on cell's membrane. Therefore, the ligand has to first cross lipid membrane, and then once it's inside, it can then bind to the receptor. Now, the activated ligand receptor complex can move into the nucleus, bind to DNA, and regulate gene expression, ultimately leading to synthesis of specific proteins. Now, let's briefly talk about life cycle of receptors. So, each cell's DNA contains code that's used to synthesize proteins from which different receptors are assembled. Once assembled, the receptors get embedded into the cell membrane and can receive and respond to signaling molecules. But now, let me ask you, what happens if cells receive too much stimulation, which can potentially damage the cell? Well, fortunately for us, cells have the ability to downregulate receptors, meaning they can take them out of the membrane and recycle them, which leads to fewer number of expressed receptors and thus decreased sensitivity to signaling molecules. On the other hand, let me ask you, what happens if most of cell's receptors get blocked and cell receives very weak signals? Well, in that case, cells have the ability to upregulate their receptors, which means that more receptors can get inserted into the membrane, thus increasing cell sensitivity to signaling molecules. Now, let's look at the example where our signaling molecule is actually some kind of a drug. So as the concentration of that drug increases, its pharmacologic effect also increases until we reach the point at which all the receptors are occupied. If we plot this, we can determine EC50 from the graph. EC50 is simply the concentration of a drug that produces 50% of the maximal effect, and it tells us how potent the drug is. So let's call this curve drug A. Now, if we can graph dose response for a different drug, let's say drug B, which yields different curve, we can now easily tell that of the two, drug A is more potent because it has lower EC50. Simply, less drug is needed to get half of the max response. Now, what else can we see on this graph? It looks like drug B doesn't even reach the same level of pharmacological effect as drug A, and this is due to its efficacy. So maximal efficacy of each drug is represented by E max, at which we assume that all the receptors are occupied by the drug, and higher concentrations of it don't produce larger effect. So in this example, drug A is not only more potent, but also more efficacious. Now let's talk about intrinsic activity of drugs. So intrinsic activity refers to ability of a drug to produce maximal effect. So if a drug binds to a receptor and is able to produce maximal effect that is comparable to effect produced by our body's own endogenous ligand, we call it a full agonist. Now, let's say about 15% of receptors show some kind of activity when there is no agonist around. This is what we call basal activity. So again, in presence of full agonists, we would see maximal effect. Next, if we have agonist that is unable to produce maximal effect, even if it occupies all the receptors, we call it partial agonist. Lastly, if we have an agent that binds to the receptors and instead of activating them, it stabilizes receptors in their inactive form, we call it inverse agonist. And this is because it simply eliminates basal activity. On the other side of the spectrum, we have antagonist, which refers to a ligand that can bind to a receptor and block it, thus reducing agonist activity. So if we have both agonist and antagonist, that can bind to the same site on the receptor, they will compete for that site. Therefore, antagonist that binds and prevents agonist from binding, it's called competitive antagonist. The characteristic of competitive antagonist is that it shifts dose response curve of the agonist to the right. So for example, if agonist is some drug in the presence of competitive antagonist, we need higher concentrations of that drug to get half of the max effect, which is EC50, so potency is reduced. Now, some antagonists can form covalent bonds with active site on the receptor and thus irreversibly block it. This is non-competitive process because irreversible antagonists can't be displaced by agonists, which leads to reduction in maximal effect, which is Emax, and thus reduction in agonist efficacy. Another type of antagonist is called allosteric antagonist. Unlike the other ones, these bind to the site different from agonist binding site and induce conformational change which prevents agonists from activating the receptor. And just like with irreversible antagonists, the allosteric ones also cause reduction in Emax, but no change in EC50. The last important concept in pharmacodynamics that I would like to talk about is therapeutic index. Our bodies are very complex, therefore not everyone will experience the same exact effect from the same dose of a drug. This is why we came up with therapeutic index, as it helps us to measure the relative safety of a drug for a particular treatment. So therapeutic index is simply the ratio of the dose of a drug that produces toxicity in 50% of the population to the dose of a drug that produces effective response in 50% of the population. So if we were to graph it, we would need to determine the curve that represents positive therapeutic response and the curve that represents negative toxic response. So in this example, after obtaining data from the population, we can determine dose of a drug at which we observed effective response in half of the population, which is our ED50, and we can also determine dose of a drug at which we observed toxic response in half of the population, which is our TD50. Now, having all that on the graph, we can easily determine therapeutic index, which is this range of doses at which a drug provides benefits without causing major toxicity. And with that, I wanted to thank you for watching and I hope you enjoyed this video.